Okay guys, we've been learning about diffusion, so let's go just a little bit deeper now. Everything we've talked about so far is something called passive diffusion. That is, we get diffusion directly across a cell membrane, traveling down a concentration gradient like I've got here. So here's our lipid bilayer of the cell membrane, and materials on the high concentration side can actually squeeze their way between these phospholipids and into the cell. Now, what happens then if our particles are too big to squeeze between those phospholipids? In that case, the cell has another strategy. That strategy is called facilitated diffusion. In facilitated diffusion, it's a little bit different than passive diffusion in that materials have to pass through special carrier proteins. All right, so that's how they get across the membrane. The particles are too big to squeeze between the phospholipids, so instead they bounce off. In this case, these could represent molecules of glucose. So when they bounce off, though, they still are trying to pass from this high concentration down to a low concentration. They just have to do it through specialized carrier proteins that act like a channel. This one, in this example, happens to be an integral protein, or what we would just call a transport protein. And so again, materials can still get across that membrane. They're just going to have to use carrier proteins to do it. But other than that, the process is exactly the same. Materials diffuse from an area of high concentration to an area of low concentration.